All right, so last video for the senses where we're going to get into hearing and equilibrium. These are together since they are both going to involve the ear. So we're going to talk about those senses. We'll look at the anatomy of the vestibule. We'll look at hair cells for equilibrium, semicircular canals for rotational equilibrium, and then get into those neural pathways as well as get into all of the pieces that are involved with hearing, and talk about how we transmit sound waves into an action potential. So when we say hearing, what we're talking about is the transduction of sound vibrations. So vibrations in the air, basically, by mechanoreceptors. So we're using mechanoreceptors or hair cells in the cochlea. We're using the same type of things, mechanoreceptors, in the vestibule and semicircular canals with equilibrium. But with equilibrium, we're looking at the transduction of fluid movement, right? So sound vibrations are going to be converted into fluid movements as well for hearing. Equilibrium, there is no air, there is no sound vibration, it's just straight fluid movement. So let's go ahead and just remind ourselves about the ear's anatomy. I'm going to go through this relatively quickly, but if you need to pause here, by all means. We can break the ear into 3 different regions: the external ear, the middle ear, and the inner ear. I like to explain the external ear as like the part you can touch with your fingers, whereas the middle ear is what you don't want to touch with a Q-tip. And the inner ear, well, if you've touched the inner ear, you've gone through bone, so you have a lot of problems at that point. So the external ear is going to consist of the auricle, also sometimes called the pinna, and the external auditory canal. The middle ear is where we find the ossicles, the tympanic membrane, and the eustachian tube. And then the internal ear is going to have the semicircular canals, the vestibule, and the cochlea. So let's talk about the external ear, the outer part of the ear. First, again, is the auricle, sometimes called the pinna. This is going to help capture sound waves and direct them in towards the external auditory canal. We call this the external auditory canal because it's a proper anatomical term, but you probably know it better as your ear canal. This is going to help continue to transmit those sound waves to the eardrum, also called the tympanic membrane. Within this ear canal, we have ceruminous glands. They're secreting cerumen, which is they're basically modified sweat glands, or they're similar to sweat glands, and they release earwax, and this is to help protect the canal. So you actually do need some earwax. It is protective and whatnot, so you should not have completely clear ear canals. You should have a little bit of wax in there. However, if you notice your earwax is affecting like your hearing, then there are ways to properly remove it. Q-tips are not the answer there. You should not use Q-tips because you can push them too far to the point where you can touch or damage the tympanic membrane. And most of the time a Q-tip just pushes the wax deeper rather than removes it. Instead, you should use drops that are specifically made to dissolve or soften earwax, or best case / worst case scenario, go to a doctor where they can help remove it with specialized tools or washes that are made to remove wax. Moving into the middle ear, we get to the tympanic membrane, also called the eardrum. I will use the term tympanic membrane just so that we keep it consistent. And then right next to that tympanic membrane, we have the ossicles, or the little bones. There are 3 of them, and in their order they go the malleus, the incus, and the stapes. The malleus, the incus, and the stapes are going to basically take the sound vibrations from the tympanic membrane. So sound waves are directed in through the ear canal, right through the external auditory canal. The sound waves strike the tympanic membrane like the top of a drum, and the ossicles are going to amplify those vibrations from the tympanic membrane. So they are going to take those vibrations from the tympanic membrane, they're going to pass through the 3 bones, and as they pass through to the oval window, they will be amplified so that we kind of make them even bigger, right? There's also the auditory tube, also called the eustachian tube. This extends from the middle ear down to your throat in the nasopharynx region. This is to help regulate air pressure. Because your middle ear is filled with air, not fluid, that means changes in elevation, changes in altitude can affect air pressure. If it were sealed off, then you could potentially rupture your tympanic membrane, right, your eardrum, by going in a plane, right, or going on a hike to a really high elevation. So you've probably noticed if you've ever like gone flying or whatever that you chew gum and your ears quote pop. That is the eustachian tube regulating and fixing basically the air pressure in the middle ear to make it the same and equalize it to the air pressure exerted outside in the external ear, because you want to keep those the same, right? If you have a significant difference in air pressure on one side of the tympanic membrane, right, compared to the other, then it can push on that membrane so much that you actually rupture your eardrum. So eustachian tube is your built-in protective feature against that. Then we get to the inner ear. The inner ear has the cochlea. This is like the little snail shell that translates vibrations into sound. The vestibule, which is like the snail's body or head that detects balance and equilibrium in terms of gravitation or gravitational equilibrium. And then the semicircular canals, you have 3 of them. These are kind of like the little eye stalks for the snail. They are going to be used for balance and equilibrium on the rotational side of things. And so we're going to talk about all of these. So we're going to talk about balance first, and then we'll get into hearing. So balance or equilibrium has 2 different forms, gravitational and rotational. Gravitational equilibrium is maintaining your body's position relative to the force of gravity. So think of like your up and down, right? This is going to be affected or controlled by your vestibule or detected by your vestibule, which has 2 parts, the utricle and the saccule. And then the rotational equilibrium, this is your maintaining your body's position in response to movement. You have 3 semicircular canals kind of going in an X, Y, and a Z-like fashion. So one's going like up, one's going left and right, one's going forwards and backwards. These are going to help sense when your head moves in any direction, right? So that's what the rotational equilibrium is, right? We're going to talk about, we're not going to look at the picture here just yet. We're going to talk about the same picture in a couple slides. So let's talk about the equilibrium, the gravitational equilibrium, and kind of what happens in general with these hair cells. So what is going to happen is we have these modified epithelial cells. They have these little projections. They look like little hairs that are called stereocilia. When motion occurs, these stereocilia bend. This is going to open potassium channels, which then depolarizes the hair cell. And when the hair cell gets depolarized, it's going to release a neurotransmitter to depolarize the dendrites of the vestibular, the neurons that service the vestibular nerve. If we bend the hair cells the opposite way, this is going to cause hyperpolarization. So bending one way causes depolarization, bending the other way causes hyperpolarization. And this is basically what tells us direction, right? So if we move our head one way versus another, right, that's going to cause either depolarization or hyperpolarization, and this is what's going to allow us to get direction, right? Are we looking up? Are we looking down? Are we looking left or are we moving right? So let's talk about the specific forms of equilibrium. Now, we're going to get into gravitational first. This again is in the vestibule by the utricle and saccule, and we have a few main components, right? So let's take a look at the picture here. Down at the bottom here on the left, we see there is this macula, this kind of rounded out portion, and inside of this macula, right, we see there's this otolithic membrane, right? Otolithic membrane that hair cells are being pushed or kind of projecting into. And on the, like on this or in this otolithic membrane, we have otoliths, right? These otoliths are like these little calcium-based, like stones, basically, like these little components that are stiff. And when you move your head, right, that causes the otoliths to move, right, within this otolithic membrane, right? So like this guy here, he's looking straight ahead, everything's nice and up and right. But when he tilts his head forward, we see that otolithic membrane shifts because of gravity, and the otoliths shift with it. That causes the otolithic membrane to push down on the hair cells, and that pushes the stereocilia, bending them, and that's what causes the receptor potential, so we can send a signal. So the hair cells, right, they bend, right, their stereocilia bend in response to the force of gravity being placed on them, and so we're going to then synapse with the neurons in the vestibular branch of the vestibular cochlear nerve, and then have that impulse travel to the cerebellum as well as the vestibular area of the cortex via the thalamus. But the big thing here is just looking at this and kind of understanding what these parts are doing. Like the macula is kind of keeping everything encased, right? We have hair cells with stereocilia that extend up into this otolithic membrane, and then otoliths that are on top, and that those otoliths are going to move, right, with gravity, and that's with the otolithic membrane, that's going to cause those hair cells to bend. And when those hair cells bend, that's how we send a signal to the neurons. Rotational equilibrium, this is, again, you have 3 of these semicircular canals. They're all at right angles, so you have like one that's like up and down, one that's left and right, one that faces forward and backwards. That gives you kind of 3 dimensions. And at the base of each semicircular canal, we have what's an ampulla. And so here's our ampulla here, down here in this picture. The person's head is still, and so you see everything is nice and still, right? But in this ampulla, we have this cupula, right? We see those hair cells with their stereocilia kind of extending up through that cupula, right? And inside here, we have fluid called endolymph, right? As a person's head moves, right, like they rotate their head, they turn it to the left or the right, right? This entire cupula is going to bend opposite, right? So this person's head is turning this way, right? This whole thing's going to bend in the opposite direction of the rotation. And as it bends, the hair cells will also bend, right? When the hair cells bend, we go through that same process, right, where we depolarize or hyperpolarize depending on how we bent, and then that nerve impulse that'll cause us to release neurotransmitters to communicate with the neurons, which will then stimulate a nerve impulse along the vestibular branch of the vestibular cochlear nerve. Same thing, we're traveling along the cerebellum to the vestibular area of the cortex via the thalamus. Then we get into hearing, right? Hearing, I'm going to walk us through this picture here because I really like it. It kind of keeps things relatively simple but explains things very well rather than having you just read a bunch of bullet points. So we're going to walk through what happens, and then we'll talk about the specific organs and the hair cells and all that. But number 1, sound waves, right? Basically, you have a high air pressure and low air pressure. And sound waves are like, you're changing between those two, between high and low pressure. If we look at what a wavelength is with that, we see that the wavelength is going from high to low and back up to high. The amount of time, right, that it takes, right, for you to go from high to low to high, that's a wavelength, right? That's the frequency of the wavelength. Then you have amplitude, which is how high that point goes, right? The high point goes. So we have these sound waves, right, that are basically just little changes in air pressure. They're going to be collected through the ear, right? They're going to travel through the ear canal, through the external auditory canal, and they're going to strike the tympanic membrane. And just like if you were to strike a drum, right, you would see, like, if you really slowed it down, you would see the drum vibrate, right, in response to you striking it. Same thing here. The tympanic membrane is going to vibrate in response to being struck by sound waves. So far, everything is happening through air. Once the tympanic membrane has been struck, the ossicles, right, malleus, incus, and stapes are going to amplify that, right, those vibrations. So make those vibrations even stronger. Right, and we see that the stapes is going to be at this oval window and it's going to strike the oval window. Right, we strike the oval window right up against and we're going to send a wave of fluid. So at this point, from the middle ear to the inner ear, we're converting from an air-filled space to a fluid-filled space. So now we are inside of the cochlea, right? We're in the cochlea, and we see that these vibrations are sending a wave in the fluids inside the vestibuli. We have the scala vestibuli here. We have the scala tympani down here. how frequent they are, right, is going to tell us how frequent these waves happen, right? And then how loud it is going to tell us how big these waves are, right? So the pressure there, though, from these waves, right, as these waves pass through, is going to put pressure on the membrane of the cochlear duct, right? This is going to cause the hair cells in the basilar membrane to vibrate. And it's those vibrations, those bends, those changes in the shape or motion of the hair cells that is going to be converted to sound for us. So pressure, right, again, the oval window is struck by the stapes, and this causes pressure waves in the fluid inside the scala tympani. So again, it travels through the scala vestibuli, and then it will wrap around down to the scala tympani. As it goes to the scala tympani, the basilar membrane, this membrane right here, is going to vibrate. This is going to push stereocilia, right, against the tectorial membrane. This is the tectorial membrane over the top. So we're kind of pushing it up, right? Kind of squishing those stereocilia of the hair cells up against the tectorial membrane. And as we bend those stereocilia, this is what triggers that receptor potential in the hair cells. We release a neurotransmitter. That neurotransmitter is picked up again by sensory neurons, which then release an action potential. So if we take a closer look at the spiral organ, or the organ of Corti, this is where we have that basilar membrane, the hair cells with the stereocilia and the tectorial membrane. Those stereocilia are found in bundles, right? So we see them like in these little groups, and they're going to bend, right, as they're pushed up against the tectorial membrane. They're going to bend in response to the pressure waves, right? And when we bend these stereocilia, we're going to see something similar in that potassium channels open. This causes depolarization of the hair cells, and then we, communicate via neurotransmitters from the hair cells to the sensory neurons. Sensory neurons activate their action potential, and we send that along the vestibular cochlear nerve. So those cochlear nerve fibers, they're going to form the cochlear branch of the vestibular cochlear nerve. This is going to synapse along the medulla oblongata. So if we look here, right, here's our cochlea, here's our cochlear nerve, right? We see that we're entering in, synapsing in the medulla, and then traveling up through the medulla, through the pons, through the midbrain, up to the thalamus, and then from the thalamus, right, we will synapse towards the primary auditory area in the temporal lobe. So how do we determine volume and how high pitch or low pitch, right, frequency? How do we determine that? So we need to first understand sound waves. Sound waves are just the result from alternating between high pressure and low pressure of air molecules. And that going from high to low to high is what we call a wavelength. We have the wavelength, which is again the distance between the peaks, but we also have the amplitude, which is how high the peaks go. The higher the amplitude, the louder the sound, right? Because this is going to bend those hair cells even more, right? That's how the hair cells detect loudness, is that the higher the amplitude, right, the harder, like the bigger that push, the more those hair cells, their stereocilia get bent. And then frequency of the wavelength, how close the peaks are versus how far away, is pitch. The higher the frequency, meaning the closer those peaks are together, the higher the pitch, right? The further apart these peaks are, that's the lower frequency, the lower the pitch, right? So high pitch would be like someone who can sing soprano. I can't sing, so I'm not going to. Versus someone, maybe they're baritone, they have a lower pitch, that means lower frequency. High pitch sounds and low pitch sounds will also differ in where they kind of trigger that basilar membrane more, where they cause that vibration. So here we've taken the cochlea, we've kind of unrolled it, right? We've expanded it out. And we see that the high pitch ones, the high frequency, see how close together those peaks are, right? High frequency, so high pitch, we see those ones are going to cause the basilar membrane to vibrate really close. Whereas as the pitch goes down, right, as it lowers, the frequency becomes medium or low. We see those peaks get further and further apart, right? We see that the basilar membrane vibrates further and further down the line. So this is how not only can we tell volume, how loud something is, but we can also tell the difference between someone who is singing, let's say, in falsetto compared to someone who's singing in a very low baritone voice. And of course, with the ear, there are things that can go wrong. I'm just going to cover a couple things. One is deafness, right? This is where you have either significant or total hearing loss. Someone who is deaf, they may have complete hearing loss. They hear absolutely nothing. They may have profound hearing loss, meaning they hear some things but not enough to make sense of anything they're hearing, right? So they would have a very difficult time. Sometimes you also may hear the term hard of hearing for someone who has significant hearing loss. This can be something someone is born with. This can be something that happens later in life. It can be in response to an illness, especially in childhood, such as certain infections can lead to deafness if they happen at certain times during childhood. This can be caused by a couple things. It can be caused by impairment of the cochlear or the cochlear branch of the nerve. So basically there is the inner ear and the nerve is the issue. Or it could be because the middle ear does not work. It's transmitting sounds to the cochlea, right? So it could be either way. So either conduction, right, or it's neural, right? The neural pathway is damaged. Otitis media, this is an infection of the middle ear. This is what you would know of as an ear infection, something that children tend to get when they're little. This is generally caused by bacteria, and this is characterized by pain, right? The child may be tired, fatigued, they may have a fever, and then we see the eardrum will actually be bulging or in red. If it is really serious, then it could potentially rupture, which would be very painful. This can be treated, right, by just going to a doctor and getting medications. Tinnitus, this is where you hear sound when there is no sound. A lot of people will describe it as a ringing in the ears, although it doesn't always have to be a ringing. As someone who is in the military, I actually have tinnitus. Sometimes it's actually technically tinnitus, but everyone calls it tinnitus, and so I will too. Tinnitus, again, is just sound where there is no sound. It can be very quiet, but for some people it can be very disruptive in their life. Some people describe it as a ringing or a humming. In my case, I have ringing, but I also have a staticky sound sometimes. We don't actually fully know what can cause tinnitus for some cases. It can be caused by loud noise exposure. So like in my case, I was military, and so my time there exposed me to a lot of loud noises without proper protection. And so that's most likely the cause of mine, but for some people we don't always know what caused it. And then vertigo. This is a loss of equilibrium, right? Someone is standing still and yet they feel like they just got off a tilt-a-whirl. They're dizzy, they're sweating, they're nauseous, they're vomiting, right? They feel like they're spinning, right? Even though they haven't done anything to cause that loss of equilibrium. That can be very disorienting and disconcerting for a person as well.